mengumumkan akan masuk diketuai oleh Timbalan Naib Chancellor Hal Ehwal Akademik dan Antarabangsa dan para profesor. Hadirin mohon berdiri. Hadirin dipersilakan duduk. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Yang berbahagia, Profesor Datuk Insinyur Dr. Muhammad Marzuki Mustafa, Timbalan Naib Chancellor Hal Ehwal Akademik dan Antarabangsa mewakili Naib Chancellor Universiti Kebangsaan Malaysia. Yang berbahagia, Timbalan Naib Chancellor. Yang berbahagia, Profesor Dr. Muhammad Kasim, Dekan Fakulti Sains dan Teknologi, para dekan dan pengarah, rakan-rakan profesor, Profesor Madya dan pensyarah, Tif-Tif kehormat dan hadirin-hadirat yang dimuliakan sekalian. Bagi memohon keberkatan majlis ini, dijemput Encik Hasan Omar untuk membacakan doa. Majlis dengan segala hormatnya, mempersilakan.
Bismillahirrahmanirrahim. Al-Fatihah. A'udzubillahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Arrahmanirrahim. Maliki yaumiddin. إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين. بسم الله الرحمن الرحيم. الحمد لله رب العالمين وعاقبة المتقين والصلاة والسلام على سيدنا محمد وعلى آله وأصحابه أجمعين. Ya Allah, Ya Ghaffar, Ya Tuhan kami, ampunkanlah dosa kami, dosa kedua ibu bapa kami, dosa guru-guru kami, dan dosa seluruh umat Islam sama ada yang masih hidup atau yang telah meninggal dunia. Allahumma anta azizul jalil. Sesungguhnya kami pada petang ini ingin merafakkan kesyukuran dan pujian terhadap-Mu atas kurniaan-Mu yang tidak dinilai. Ya Allah, Ya Ghaniyul Ya Hamid, sesungguhnya kurniakanlah kepada kami kekayaan ilmu pengetahuan dan hiasilah diri kami dengan sifat lemah lembut, dan kurniakanlah kami dengan ketakwaan, dan indahkanlah diri kami dengan kesihatan dan kesejahteraan. Ya Allah, Ya Hayyum Ya Qayyum, justru itu jadikanlah majlis Syarahan Perdana Prof Dr. Wickneswari Ratnam yang bersejarah dan penuh bermakna ini mendapat keberkatan dan kemuliaan dari-Mu. Satukanlah hati-hati kami, kukuhkanlah tali silaturahim antara kami, dan kurniakanlah kejayaan yang gemilang di dunia dan di akhirat atas usaha-usaha yang telah kami lakukan. Ya Allah, Ya Rahman Ya Rahim, kecemerlangan kami dalam melaksanakan tugas kami adalah di bawah kekuasaan dan kudrat-Mu. Tanpa pertolongan dan bantuan-Mu, kami tidak akan mampu mencapai kejayaan semoga amal hari ini. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azabannar. Wassallallahu ala sayyidina Muhammad wa ala alihi wa sahbihi wa sallam. Alhamdulillahirabbil alamin.
Terima kasih di atas bacaan doa sebentar tadi. Memulakan atur acara, majlis dengan sebuah hormat mempersilakan Yang berbahagia Profesor Senat Dr. Muhammad Marzuki Mustafa Timbalan dan Chancellor Universiti Kebangsaan Malaysia untuk menyampaikan pidato pengenalan. Majlis dengan segala hormatnya mempersilakan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan selamat petang. Yang berbahagia Timbalan Anak Chancellor, yang berbahagia Profesor Dr. Muhammad Kasih, Dekan Fakulti Sains dan Teknologi yang kita raikan, yang berbahagia Profesor Dr. Wickneswari Ratnam, Profesor di Fakulti Sains dan Teknologi, para Dekan dan Pengarah, para Profesor Madya, Tuan Pesara, Dewan Hormat hadirin dan hadirat yang dimuliakan sekalian. Pada petang ini kita memberi penghormatan kepada Profesor Dr. Wickneswari Ratnam dari Fakulti Sains dan Teknologi yang akan menyampaikan syarahan perdana beliau dengan tajuk Genetik dan Genomic Tumbuhan Tropika, Pelaburan untuk Masa Hadapan. Profesor Dr. Wickneswari Ratnam yang lebih mesra kita panggil sebagai Prof Wiki oleh semua orang, dilahirkan di Ladang Seremban, Negeri Sembilan pada 19 Februari 1958. Beliau merupakan anak ketiga kepada pasangan Ratnam Wilatilingam, seorang pesara guru besar Sekolah Tamil, dan Sinatilan Bani Tamdi, seorang suri rumah tangga sepenuh masa. Profesor Wiki mendapat pendidikan awal di Sekolah Rendah Convent of the Holy Infant Jesus, Seremban dari 1965 hingga 1970. Beliau kemudiannya meneruskan pengajian di peringkat menengah di sekolah yang sama dan di Sekolah Menengah St. Paul, Lobat, Negeri Sembilan dari tahun 1976 hingga 1977. Pada tahun 1978, Prof Wiki menerima tajaman beasiswa daripada Yayasan Negeri Sembilan untuk melanjutkan pengajian ke peringkat ijazah sarjana muda di Universiti Malaya dan berjaya memperolehi ijazah sarjana muda sains dengan kepujian kelas 2 atas dalam bidang genetik pada tahun 1982. Pada tahun itu juga beliau meneruskan pengajiannya di peringkat sarjana dan doktor falsafah di UM. Beliau berjaya menukarkan taraf pengajian dari MPhil ke PhD pada tahun 1984 dan mendapat ijazah PhD pada tahun 1988. Penyelidikan PhD beliau melibatkan pemahaman aktiviti biologi sebahagian teoganotin ke atas kulat yang menyebabkan penyakit tumbuhan dan kesannya kepada mikrob tanah berguna. Kajian ini dijalankan bersama kumpulan penyelidikan rentas disiplin sebahagian organating UM yang diketuai oleh Profesor Bilik Das, Fellow Academy Science. Dalam tempoh menyelesaikan pengajian PhD, beliau memulakan perkhidmatan sebagai pegawai penyelidik di Institut Penyelidikan Perhutanan Malaysia atau FRIM di Kepung pada 2 Oktober 1986. Dalam tahun yang sama, Prof Vicky menambahkan zaman bujannya dan mendirikan rumah tangga dengan Encik Mulyandi Dahlia. Ini sudah handsome gentleman. Pasangan bahagia ini dikurniakan oleh 2 orang putera, iaitu Yang Garan Mulyandi dan Mikal Mohan Kumar Mulyandi. Di sini. Pada tahun 1993, dia telah dinaikkan pangkat ke pegawai penyelidikan di FRIM dan giat menjalankan penyelidikan genetik hutan tropika. Kajian beliau ini diteruskan di UKM apabila beliau memulakan perkhidmatan UKM melalui skim pemindahan perkhidmatan terus ke jawatan Profesor Madya di Fakulti Sains Hayat pada 2 Januari 1958. Atas kecemerlangan Prof Vicky, pada 1 Oktober 2005, beliau telah dinaikkan pangkat sebagai profesor. Kajian tentang impak genetik sistem pengurusan hutan di Malaysia adalah merupakan bidang kajian yang mendapat perhatian mendalam dari beliau sehingga berjaya menghasilkan pembentukan kaedah terperinci dalam garis panduan penebangan pokok hutan asli yang dibangunkan bersama Jabatan Perhutanan Semenanjung Malaysia. Kajian yang dijalankan dalam bidang ini juga berjaya menghasilkan sebuah buku mengenai pengurusan sumber hutan tropika yang bernilai tinggi yang telah dilancarkan pada tahun 2012 oleh Ketua Pengarah Jabatan Perhutanan Semenanjung Malaysia. Bidang penyelidikan beliau tidak berhenti dengan pengurusan pokok hutan sahaja, tetapi bermula pada tahun 2000, Prof Vicky mengetui beberapa projek rentas institusi dan disiplin bagi menjalankan kajian terhadap genomik dan biak baka akasia dan padi. Hasil kajian beliau telah diterbitkan dalam lebih daripada 117 makalah jurnal di peringkat antarabangsa dan kebangsaan, 200 kertas persidangan di dalam dan di luar negara, dan diterbitkan lebih dari 14 buku ataupun bab dalam buku. Selain itu, antara hasil penyelidikan beliau yang begitu signifikan kepada universiti dan masyarakat adalah penghasilan baka baru padi dan hybrid akasia, penanda molekul bagi pengesanan klon dan penentuan komposisi genom akasia, dan pemilihan awal dalam program biak baka pokok akasia dan padi. Hasil penyelidikan yang dijalankan 3 varieti padi, iaitu 2 padi berhasil tinggi di kawasan berlainan dan 1 varieti beras merah panjang dengan indeks glisemi rendah. Daripada 6 baka baru padi telah berjaya dikomersialkan bersama syarikat pertanian di Semenanjung Malaysia, manakala hybrid akasia sedang diuji di ladang bersama satu syarikat perhutanan di Sarawak. Kecemerlangan Prof Vicky dalam bidang penyelidikan dan ketukaan beliau dalam bidang pengurusan telah berjaya memperolehi beberapa gelar penyelidikan sebagai ketua projek dan co-penyelidik daripada agensi dalam dan juga luar negara bernilai lebih daripada 100 juta ringgit Malaysia. Beliau juga telah dilantik sebagai ahli Majlis Padi dan Beras Negara sejak 2016, Fellow Akademi Sains Negara sejak 2012, Coordinator International Union of Forestry Research Organisation IUFRO Working Committee on Genetics and Silviculture of Acacia dari tahun 15 hingga 2019, penyelidik adjun di Universitas Tasmania 2014-2017, penyunting bersekutu Jurnal Tree Genetics dan Genome sejak 2013, dan Koordinator IUFRO Working Party on Population Conservation and Ecology Genetic dari 2010 hingga 2014. Sepanjang kejayaannya, Prof Wiki telah menerima beberapa anugerah penyidikan dan penerbitan termasuk Inovasi Penyidikan Terbaik tahun 2013, Fellow Pelawat Saintis di Cooperative Research Center for Sustainability Production Forestry, University of Tasmania pada tahun 2003, Chevening Award tahun 1993, Anugerah Perkhidmatan Cemerlang pada tahun 2001 dan 2003, dan anugerah penerbitan terbaik dari pihak FRAME dan Lembaga Getah Malaysia serta UKM. Selain itu, beliau juga pernah dilantik sebagai ahli panel Science Fund dalam bidang agrikultur di MOSTI dan juga di MOA serta menjadi panel penilai bagi semua dana pendidikan, pembangunan dan perkomersialan di MOSTI. Di peringkat UKM, beliau merupakan ketua kluster Genomic dan Biologi Sistem dan timbalan pengarah niche kepelbagaian biologi dalam pembangunan bioteknologi. Dalam bidang pentadbiran, beliau pernah dilantik sebagai Ketua Jaringan Industri dan Masyarakat di Fakulti Sains dan Teknologi dari tahun 2013 hingga 2018 dan sebagai Timbalan Dekan Hejin pada Januari 2018 sebelum beliau bersara pada 19 Februari 2018. Beliau kini berkhidmat sebagai Profesor Kontrak di samping menjalankan tugas sebagai pengarah urusan Nomatek Sdn Bhd yang merupakan sebuah syarikat yang menyediakan peluang pekerjaan kepada alumni FST di samping menyediakan peluang latihan kepada pelajar dan alumni FST untuk menjadi usahawan terlatih. Hadirin dan hadirat yang saya hormati sekalian, saya dengan sukacitanya mempersilakan Profesor Dr. Wickneswari Ratnam untuk menyampaikan syarahan perdana beliau yang bertajuk Genetik dan Genomic Tumbuhan Tropika, Pelaburan untuk Masa Hadapan. Sekian, Assalamualaikum warahmatullahi wabarakatuh.
Pengenalan yang agak panjang lebar. Terima kasih kepada Timbalan Nak Chancellor. Selamat sejahtera dan juga selamat petang kepada semua. Yang berbahagia Profesor Datuk Insinyur Dr. Muhammad Marzuki bin Mustafa, Timbalan Nak Chancellor UKM, Hal Ehwal Akademik dan Antarabangsa, mewakili Nak Chancellor. Yang berbahagia Timbalan Nak Chancellor, yang berbahagia Profesor Dr. Muhammad Kasim, Dekan Fakulti Sains dan Teknologi, para dekan dan pengarah, rakan-rakan profesor, Profesor Madia, pensyarah, div-div kehormat dan juga rakan industri saya dan ahli keluarga yang saya kasihi yang hadir di sini, hadirin dan hadirat yang dimuliakan. Seperti yang diberitahu ataupun yang dimaklumkan oleh Timbalan Nak Chancellor, tajuk syarahan perdana saya hari ini adalah Genetik dan Genomic Tumbuhan Tropika. Mungkin ada di antara hadirin yang fikir-fikirkan apa itu pelaburan untuk masa hadapan dengan menjalankan penyelidikan dalam bidang genetik dan genomik tumbuhan tropika. Memang itu adalah satu pelaburan yang mesti diadakan oleh ataupun diwujudkan oleh kerajaan supaya kita dapat menikmati apa yang kita ada hari ini dan juga generasi akan datang juga boleh menikmati hasil kita. Kajian genetik dan genomik boleh dijalankan pada tahap penyelidikan fundamental, tahap penyelidikan gunaan dan juga tahap penyelidikan translational yang sangat-sangat kendati pada masa ini. So, maklumat genetik dan genomik yang dijana ini daripada penyelidikan merupakan pelaburan dalam pembangunan kaedah-kaedah yang berasaskan genetik dan juga produk-produk yang boleh dihasilkan yang bernilai ini. Seterusnya, izinkan saya meneruskan syarahan ini dalam bahasa Inggeris. Teks dalam bahasa Malaysia boleh didapati selepas saja syarahan ini. Okey, ini adalah untuk menghargai kehadiran beberapa orang warga asing untuk memahami ucapan saya ini. Tropical plant genetics and genomics. This is the area of research that I ventured into 30 years ago. My research actually started in the Forest Research Institute of Malaysia, where we looked at genetic assessment or assessment of genetic diversity of tropical forest species with the focus to understand sustainability of forest management or what forest management practices undertaken by the Malaysian government. So there are 3 highlights which we like to focus and results from my group, besides many other researches that I have conducted with my team on tropical plant genetics and genomics. So first one is the assessment of genetic diversity. I think members from PRIM are here, and I think one of my first students is also here, Dr. Lee. Yeah? Okay. The second aspect that I would like to share during this lecture is the development of genomic resources for acacia breeding. We can't rely on natural forests for its source of timber, so plantation becomes very important when we want to reduce the pressure on the natural forest. So one of the species that was selected for plantation way back in the 1980s was acacia. And so we looked at how we can improve breeding materials that can be used in plantation through genomic research. So I will share the outcome with that in the second part. The final part is actually a more something that I was interested in, to explore how genetic techniques can be utilized to improve rice. Because rice is so dear to us. We eat it every day. Some of us eat it 2 meals a day. So we need to make sure that rice production is sufficient for the Malaysians, besides being able to export in the future. So we have also interest to export specialty rice in the future or when we have it available. So I will share with you our work that we did on varietal improvement of rice using molecular techniques. So to start to give a understanding of what genetic diversity is about, and the value of genetic diversity and why we have to do all these measurements and monitoring of diversity of so many different species. So what we know now is, I think everybody understands sperms and eggs, that's for animals or humans, and seeds or reproductive cells. They actually serve as genetic bridge between successive generation of organisms. So if you want to make sure you have continuity of your generation, you have to make sure you have good quality sperms and eggs, or seeds, or reproductive cells. And what is in these cells that make it important? So within these cells lie a reservoir of genetic options, or alleles, for each trait that we see today, which we refer to as phenotypic traits, or phenotypic traits, which constitute actually the raw hereditary material of natural selection. So it's very important to maintain diversity, or the processes that govern diversity, and this is a powerful strategy for a species to survive, including us. So genetic diversity then becomes the basis of all diversity, or becomes the basis of biodiversity, which we are more familiar with instead of genetic diversity. So if you want to know what is biodiversity, biodiversity can be described as 3 different components or aspects. You can refer to it as compositional diversity, relating to the genes, the individuals, the species, different types of species, and the habitats. We have different types of habitats. So that's compositional diversity, which is biodiversity, or functional diversity, which relates to the processes that govern this diversity, which is the ecological processes, for example, predation, mutualism, and also parasitism. Because we have some plants that live on other plants. They don't make their own food for their energy. And the other aspect of biodiversity can relate to a structural diversity, because all species or all organisms don't occur at the same time or in the same space. They have different spaces within the ecosystem where they occupy. So some are always found in the forest floor, some on the tree trunks, some on the canopy. So we have distribution of diversity is in space within ecosystem, and it changes. It's not static, it's dynamic. So diversity is dynamic. So it's important to conserve diversity. In order to conserve diversity, it's important to make sure that the processes that govern diversity is conserved. As far as Peninsular Malaysia or Malaysia is concerned, we have all this diversity resides in forest reserves, or where you have forested areas, or we have plants. So in Malaysia, we have designated forest management areas, and 2 main forest management areas are the protected forest areas, which comprise water catchment areas, virgin jungle reserves, wildlife sanctuaries. And these are untouched, no logging, no poaching, or no hunting, okay? Or no collection of wild resources from there for commercial use. We have what we know as production forests, and there are 3 types. The lowland forest, which we are losing most of it, hill forest, and also state-land forest, which comes under the jurisdiction of the state government. In these forests, logging is permitted, and quite often in the early days they do clear-cut felling, but now it's more, they have resorted to selective felling. Okay. So this is protection of catchment areas, very important. You want clean streams and rivers, and this will guarantee you water quality. And every now and then we hear in the newspaper or we read it that we have pollution taking place and what are the causes. So it's more enforcement that needs to be put in place beside all these guidelines. So protection of catchment areas is very important for water quality. We also need to protect forest areas, whether they are virgin jungle reserves, high conservation value forests. Some forests are rich in very highly commercial species or species of ecological importance. Wildlife sanctuaries where our mammals are there. And conservation not only confined to the flora, but also fauna. And when we talk about flora and fauna, it means you have to conserve all living organisms in that ecosystem, including the fungi or mushrooms, as these play a very important role in nutrient cycling. When it comes to production forests and state-land forests, there is a lot of argument whether we can commercially log these forests and yet make sure that the ecosystem is retains back or recovers to the natural state. Is it possible or not possible? But we say we need to do sustainable forest management or sustainably manage natural forests. Can we do it? So commercial logging, does it jive with sustainable management of natural forests? So this is the area that we investigated after having understood the genetic diversity of the many tropical tree species. We looked at the impact of logging and other forms of disturbance on the genetic diversity of both the commercial and non-commercial forest species. So for this, my team, this one I think way back in 1995 onwards, we worked on this and we came up with some solutions. So we not only looked at genetic diversity, but we also looked at the species, the demography of the habitats that we were studying. So as far as logging is concerned, there seems to be minimal effect on the species diversity. Here we show you the graphs which we have plotted across different sizes of the sampling area, Q1, Q2, Q3, Q4, Q5, bigger area. Each quadrant is about 20 meter by 20 meter square. And this was sampled in a forest area which was not locked, the green triangle. And then you have the magenta and blue, which is the forest area that was regenerated but neighboring to the unlocked forest. So here, as far as species is concerned, we are very well endowed with many species, and as you increase the area of sampling, the number of species also increases. So we are actually sitting in a mega biodiversity country. So we are very well endowed, and logging doesn't seem to affect the species diversity, but the type of species is affected. Okay, the composition of species is affected. We also looked at the impact of logging on the trees that we had sampled for our genetic assessment. So as far as the area occupied by the trees, we found that logging does do a damage. What we see is a smaller diameter trees, less than 5 cm, 5 cm to 15 cm, they decrease drastically, and also trees that are 45 cm DBH, diameter at breast height, they also decrease drastically due to logging. So we lose the big trees, which is intended, but the logging area, the forest area that we studied, the adolescent tree, which is about 30 to 45 cm, those are maintained. So these are the trees that will be logged in the next rotation in about 30 years. So as far as basal area is concerned, we find reduction for the smaller diameter classes of the tree species. When it comes to density of trees, there is also a decrease, but the smaller diameter trees that we saw change drastically for the basal area, there are increasing number of those trees or the size classes. So the smaller trees, which are about 5 cm, less than 5 cm diameter at breast height, you have increase in the number of those type of trees, which is a good sign to show that regeneration is taking place when there is logging, because there is gaps created in the forest which encourages seeds or germination of seedlings which are require light. Okay? So that's a good sign. But when it comes to diversity measurements, where we use a number of different types of molecular markers, the adult trees, that is the trees that was logged, the reduction is very high, and followed by seedlings, and then saplings are the least reduced in terms of diversity. So that means that we have a savior, that is the saplings in the forest, which need to be taken care of when we do the post-silvicultural treatment after logging. So these will ensure good healthy trees for logging in future. So we know that genetic viability, there are many factors that influence the genetic viability from our studies. We found that the mating system of the species can affect the viability, whether it's a species that produces seeds through crossing or through self-pollination or apomixis. And the other important aspect is the reproductive success of trees. It's important to have flowering and also seed production of trees success very consistently for different years, to make sure that it's always seeds being produced, which can be later stored in the gene banks, the seed floss gene bank for germination. So germination, that means that successful regeneration of seedlings is also very important to ensure viability, and the survival of the saplings is very much dependent on the post-silvicultural treatment. So these are very complex kind of parameters or systems that we assess with our genetic and also demographic techniques. But simpler verifiers of this viability will be, for a forester, will be to look at the tree density after logging, after a few years of logging, and also look at the sapling growth. If there is sufficient sapling growth after logging event in the next 3 to 4 years, that means the forest can regenerate well. So what are the take-home messages from our study, which was over 15 or almost 20 years? We need to maintain appropriate tree density for each species if you want to have sustainable management of the forest. And optimal tree density can be determined from the genetic data and information on the main pollinators, flowering phenology, and synchrony of individual species. I think this is very important. And species and ecosystem diversity are essential for successful reproduction of tree species. So the other important take-home message from our study is we have to make sure that the young saplings are well taken care of so that they are not damaged extensively. So this will then make sure that the forest can regenerate and give you the the return for loggers in the future, and also at the same time ensure all the water quality and air quality that we are so much in need of. All this work is actually summarized or explained in detail in this book, Managing the Future of Southeast Asian's Valuable Tropical Rainforest. And we made it simple so that any forester can read this book and use it in managing their forest. So this book has got many authors besides myself, which I think there was a video on this before all of you entered, or some of you have seen this. So we have a final chapter which is on synthesis and options for sustainable management, jointly written together with Dr. Shahrudin and myself. So we want to translate this into Bahasa Malaysia so that we can use it with the forest department, or foresters can use it. Okay, the second part of my lecture today will be on acacia genomics and breeding, and how genomics research or genetics research can help address some of the major challenges in acacia plantation forestry. So this is a picture of acacia hybrids planted in Sigamat under Prim. Okay? Prim has got a station luar penyelidikan, which they where we have this land available to plant and to evaluate the success of this hybrid. So this is in Sigamat. Besides Sigamat, we also have planted this material in East Malaysia, in Sarawak. Okay, when you talk about acacia, there are two species that we have chosen. Besides the other important widely planted acacia species. Most of acacias originate from Australia, some also from Africa and Papua New Guinea, and also parts of Indonesia. So the most widely planted acacia species is actually Acacia mentia, planted for pulpwood. So pulpwood is for making paper, all this paper. So the more paper we use, the more trees we have to destroy. So pulpwood is important primary use, and we have close to 1.5 million hectares planted globally. And the other species is Acacia crassicata, planted in about 330, Acacia auriculiformis 170, and two other temperate acacia species, Acacia saligna and Acacia menzii, planted over 500,000 hectares globally. But the main uses are for pulpwood, Acacia auriculiformis, mainly planted in Vietnam and India for solid wood. And now I see in the furniture stores, we have acacia wood, furniture from acacia wood already been utilized because we are losing some of the other more timber species which are from the natural forests. So we have plantations sourced wood for making furniture now. So just to give you an idea how this acacia, flower, pot, leaf and trunk look like, auriculiformes, it's got a sickle cell shape of the leaf. The flowers is a bottle. I think this plant is everywhere. It was introduced into Malaysia as the avenue tree, Acacia auriculiformes, planted by the roadsides. It's a legume. It produces pods, and then the flowers are bottle brush, about yellow in color, bright yellow. Acacia mendium is a pale yellow, broader leaf, but the pod is about the same, and the hybrids in between. So here we're trying to do breeding for the hybrid to try to breed for complementary traits from the two species. This is how the seeds look like. I'm sure you all have seen it. Auriculiformes has larger seeds with long curled areoles, whereas mendium has smaller with a compact areole. And if you look closer under a microscope, the ovary of auriculiformes has got 16 ovules, and pollen, which forms, it's not single pollen grain, it forms a polyad, it's also got 16. So when this, what do you call it, hybrid, when they fertilize to produce the seeds can have different genotypes. Okay, that's the thing. So this is about the mating system, and Acacia auriculiformes and Acacia mendium are mostly highly cross-pollinated species. So the uses of acacia, having understood why we want to plant acacia, so it's got many uses besides source of timber, fiber, which is pulpwood, solid wood products, also tannin. It can also be used as a food, especially in Africa, fodder for animals, and also fuel wood. It's also a good bioenergy crop for fuel wood. Can be used for production of biofuel too because of this short rotation crop. For site amelioration, because it's a legume, it can be, if you have degraded areas, it can make the soil more fertile. It's used for biomass, for energy generation, and also it's the fragrance and also flower industry as a horticultural plant. So what are the challenges? There are many challenges. If you have foresters here, they will know, they will tell you that, oh, don't plant acacia. It's got heart rot. It's got many diseases. So when we, as geneticists, we say you need to use improved materials if you want to introduce to plantation. So that's where genetics and genomics come into play. And some of the issues that we face is how to get good yield, volume, yield, good stem form, good site adaptation, resistant to diseases, and also have stability to wind stability by modulating or tailoring the crown shape. And also the wood properties is good for soiling or good for veneer, and also have the ability to be cloned, cloning potential through micropropagation and stem cuttings. And another aspect that we studied was on lignin, because lignin is comes to about, can range from 15 to 30% of the biomass is lignin, the balance is cellulose. So if you reduce the lignin, then you can have better quality paper and also better pulp yield. So the research that we did was multi-pronged, looking at the volume yield, looking at the stem form, looking at cloning potential, and also looking at getting reduced lignin in this improved material. So far in the acacia species, only one hybrid has been successfully produced, which is the Acacia mendium crossed with Acacia auriculiformes. All other hybrids are not so successful or very difficult to produce them. So now I'm going a bit technical. So the genomic approaches that were undertaken by my group was to look at how we can discover genes for certain of the traits that we want to improve. traits related to biomass, traits related to lignin content and composition, and also traits related to the fiber length. So the strategy that we used was QTL mapping, where we had to develop mapping populations. And mapping populations required us to develop genome-wide markers, which can be used in mapping, and then constructing the maps, and then finding the association between the markers and the traits that we want to improve. So this is the development of the mapping population that we undertook. My group started in 2000. We did 2 processes together with PRIM and other agencies. 2 processes were conducted to produce the F1 hybrids, and we used different types of markers, microsatellite markers, or we call it SSR markers, and also ESSR markers and CAPs. And these were not sufficient for us to do the mapping, but we were able to produce all the mapping population. So we went into developing DNA sequence-based markers, which is called single nucleotide polymorphism, using sequencing technology of the transcriptome. So we sequenced the parents of the hybrids, AA3, AM22, AA6, AM20. We sequenced them, the transcriptome, and then we use this to identify the SNPs for our mapping studies. So these SNPs became very useful for clone identification. So the SNPs now can become made into a chip to identify clones. Even though we had many SNPs, we found that 61 SNPs is enough to identify all the different individuals in the mapping population. So I show you here a panel showing you the different colors for the different genomes. Okay? So you get the same pattern even if you use a larger number of SNPs compared to a smaller number. And this is the map that we generated for the acacia. Acacia is a haploid number of 13, chromosome number 26, so you got 13 linkage groups. And this is the map that we got based on 402 SNP foci. And these are the details, which you can read in all the publications, with a minimum marker distance of 0.6 centimeter. Okay. So the phenotypic evaluation was conducted because we had to put the material in the field, and we did all the measurements together with the PRIM team. And then we also have the Vietnamese here who came to look at our plantation material. And they are also very advanced in terms of breeding of acacia. So they are interested to look at our mapping populations now, and there is some discussion on using these mapping populations for further work. So this was a phenotypic evaluation. There is definitely increase in the diameter at breast height, which is one of the key parameters to measure when you want to select materials for plantation. So that is every year there is an increment if you look at it, both for both populations and in different locations. So it's very promising, and right now we have selected about 5 promising hybrid clones which can be used in multi-location trials. So the QTLs that were detected for DBH, or diameter at breast height, we had 3 QTLs, for biomass 5, and the genes underlying these QTLs were related to stress response, disease resistance, and also growth hormones. For plant height also we found 6 QTLs, and with 2 genes related to growth, to plant height and also growth. But for wound density, even though we detected 9 QTLs, the genes had unknown functions, so which means we need to do more work to understand that. So besides doing the QTL mapping to find the linkage between the trait and the gene, we also went on to look at the genetic basis of what we see, genetic basis, and also the functionality of the genes that we discovered. So for this, we used 3 different approaches, which is transcriptome sequencing, small RNA or microRNA sequencing, and also functional analysis, which was done by my students. And from the transcriptome genome sequencing, we were able to identify all the genes involved in the lignin biosynthesis pathway. And here you show you the number of isoforms that we detected. And details of it are in the publications as given here. So the key enzymes or genes is CoA-OMT, that's one of the key, together with CCR. So I think my student, you will remember this slide definitely. Okay, so the gene-based SNPs that we detected, which was part of the PhD student of my student is here. More questions you can ask us. So we detected both synonymous and synonymous mutations. So need more work to be done to really understand whether these genes will affect the phenotype. So that's important. So what's important is we got some markers. Okay. This student, I think, is watching me from Kuching, who did the function analysis. So the genes, we want to know whether it's real. So we did transformation experiments to show that actually these genes have a function. So they can reduce the lignin in the woody tissue. So this was demonstrated by puncturing. And Ong is not here, but this was the work that was done trying to understand whether there are small microRNAs which are very short length sequences which can influence the function of genes. And we found out that there are a few microRNAs that actually can target the transcription factors of genes that are regulating the lignin biosynthesis pathway. For your information, there is a big difference between compression wood and tension wood. So compression wood will have higher lignin content compared to tension wood has got higher cellulose content. So this was demonstrated. You can see all the microRNAs are highly expressed in the tension wood. That means the lignin is downregulated. So that one of the key microRNAs here that we found that need to be done explored further, which I think I have a collaborator from Taylor's who is working on that. microRNA 256, it seems to be highly expressed in the tension wood and may be a very important microRNA to be selected for if you want to have less lignin in the biomass. And this will be very useful in the paper and pulp industry where they can get higher pulp yield. So the last part of my talk, which is more relevant to everyone and easier to understand, is rice. So how do we do varietal improvement of rice? Using molecular technique. So we don't do molecular technique by itself, but we combine the molecular technique with conventional technique. We have to do breeding to produce the mapping populations or the progenies, and then use them also in the understanding how the genes that are inherited. So rice, not necessary to explore further, to send a message that Malaysia is still lagging behind many countries in terms of rice production. So we are somewhere in the middle, Malaysia, the pink color. Number one is Vietnam. Vietnam is one produces lots of rice, and they're also one of the major exporters of rice together with Thailand. Cambodia also exports rice, but we just have enough for us to eat, or not enough actually. We produce about 70 to 72% of the rice we produce for consumption, and the balance we import. So our average yield is about 4 to 5 tons. So hope with the new varieties that were launched, which you will see shortly, we hope to increase 7 to 8 metric tons. So what we did was a new strategy, a challenge, which was not done earlier by other rice breeders here. We actually used the results from earlier work by the Chinese to see how wild rice, if crossed with cultivated rice, can improve yield. So we looked at transessive variants, that is offsprings that perform, whose performance exceed that either parent in a positive or negative way. and it generally results from interaction between genes present in the two parental types, and the way to go is to produce intercellular hybrids and do introgression. So more on these are the hidden alleles that can be identified, and these can actually contribute to increase in yield of cultivated rice. So we use references based on earlier work, and this work was started in 2001. A team of researchers, some researchers are also here. So this is a wild rice that is found in Malaysia. It looks so bad, nobody wants it, and it's black color, the seeds, and it's got a tail. And the form is also spreading. It's not the type which is compact, we call the new plant type. So this is not good, and the seeds also shatter. But when you cross it with the cultivated rice, it does wonders. So this is a crossing strategy that we did, our team, very big team. And we, end of the day, this whole flowchart here shows you the years of work, close to 15 years. It's still going on, there's so much room for improvement. But from this breeding program, we were able to detect different breeding lines with different yields and also different quality traits. So Oryza rufipogon is the one that is the first one here, wild rice. MR219 was used as the recurrent parent, and we are comparing it with Thailand red rice and also the new breeding lines, UKM-RC 9, 11, and 2. So this is, we did the map just to understand how much of the wild rice gene is in the cultivated, in the introgress lines. Okay. So the new plant variety that we got approved and registered as new plant variety is 6 different advanced breeding lines, 4 white rice and 2 red rice. And it's owned by UKM, and we Non-Matec is a company that just started to commercialize this as the exclusive licensing right. So the first variety that we commercialize, my partner is here, is a low GI, high antioxidant, tasty rice. So we have healthy tasty rice. This morning, the LPU chairman said, can you produce basmati for us, Malaysian basmati? Can we produce Malaysian basmati? We can try. Maybe it's a challenge for the younger people here to produce the Malaysian basmati. They want high quality, tasty rice. High quality, tasty rice. So everyone will get a packet of this when you leave the room afterwards. Not the 1 kg pack, but the 200 grams. We can't afford to give the 1 kg. I think my distributor will scream. Okay, the other 2 varieties that we successfully commercialized is UKM-RC2 with the registration number PBR0028, yield potential 12 tons. So farmers yields have 6 to 7, and it's got good resistance to diseases, good breeding quality, and also can tolerate submergence at the early stage. And UKM-RC8 similarly a slightly higher yield compared to UKM-RC2, but has got good breeding quality and also submergence tolerance. So this is the field evaluation team for the new rice varieties, which was initiated after we got our IP rights in 2015 to 2017. And I would like everyone to take a moment of silence for the gentleman in the big-sized gentleman in the yellow t-shirt who passed away on the 3rd of March. So it's a great loss to my team, and I would like to call up all of you to observe 1 minute of silence for this gentleman who was so enthusiastically did all the trials and coordinated my work in the northern region and also other parts. So let's do the Dua Al-Fatiha for him. I believe he is watching over us because it was his dream that one day we will produce this two varieties which will be grown at least 30% of the granary areas in Malaysia. So I hope we will live towards achieving his dream. So with that, I think there is a video presentation of the launching of these two varieties by the Minister of Agriculture and Agro-Based Industries on the 21st, and Dr. Ikhwan was there. So we will take time, just relax, watch the video.
Padi UKM RC2 dan RC8 ialah varieti terbaru hasil penyelidikan Universiti Kebangsaan Malaysia bersama Institut Penyelidikan dan Pembangunan Pertanian Mardi, dan penghasilan varieti baru ini adalah pencapaian yang memberi impak yang signifikan kepada industri penanaman padi, khususnya kepada rakyat Malaysia. Proses penghasilan varieti padi ini adalah proses kacukan antara padi tempatan melalui pendebungaan terkawal, di mana padi liar adalah sebagai induk betina, manakala padi tempatan berprestasi tinggi adalah sebagai induk jantan pada kacukan pertama. Spesies padi liar daripada alam semula jadi mempunyai kemampuan dalam meningkatkan ciri seperti ketahanan kepada banjir, penyakit, serangan perosak dan peningkatan hasil. Kajian mendapati UKM RC2 boleh menghasilkan beras putih sebanyak 12 tan per hektar dan tahan penyakit. Anak pokok RC2 menunjukkan tahap kemandirian yang tinggi walaupun terendam dalam banjir selama 14 hari. Manakala RC8 dapat menghasilkan 14 tan beras putih per hektar dengan bertahap beras hancur yang rendah. Anak pokok RC8 juga menunjukkan tahap kemandirian yang baik walaupun terendam selama 14 hari dalam banjir.
Saya mengucapkan setinggi-tinggi tahniah, terima kasih dan sejujurnya sangat terharu dengan kejayaan kombinasi beberapa agensi penting kerajaan daripada UKM, daripada Mardi dan juga Mada yang saya rasa telah mengeluarkan suatu keberhasilan yang sangat tinggi nilainya dan tidak ternilai jasanya bagi memastikan kita maju setapak lagi ke hadapan untuk kita memastikan Kementerian Pertanian dan Industri Asas Tani berupaya dan terus gagah untuk mencapai objektif pertama, teras utama yang paling penting, iaitulah membekalkan makanan yang secukupnya kepada rakyat Malaysia sebagai jaminan security makanan ataupun disebut sebagai food security, dan juga objektif kedua yang paling penting untuk kita meningkatkan pendapatan semua pesawah-pesawah kita yang telah begitu lama berjuang, memberikan jasa, memberikan segala kudrat mereka, titik peluh mereka, usia mereka untuk memastikan kita hidup dan dapat menikmati bekalan makanan beras yang cukup bagi negara kita.
Terima kasih ya. So, kita dah sampai ke akhir. Ingin memberi penghargaan kepada beberapa ahli projek dan juga co-pendidik dari dalam UKM, luar UKM, di Malaysia, luar Malaysia. So, ini adalah senarai ahli dan collaborator bagi projek Akasia. Panjanglah. Too many to say. Okay. We have the Barisan graduate students, some are here. Okay. And then also dari luar negeri, Scottish Crop Research Institute. We have from University of Tasmania, and University of Malaya is here, okay, and Frame is here. That's for the AKC project, and this is for the rice project. I have a different group of people for the rice project. Senelahnya dari UKM, dari UM, dari Monash Malaysia, dari Maldi, dari UMT, MNA, UPM, IRI, dan juga DOA. So, ramainya. And then ini adalah all the semua institusi yang terlibat dan juga termasuklah yang memberi kemudahan dari segi DNA sequencing ataupun genotyping, termasuklah rakan industri iaitu Asia Prima Resources, TAAN bagi Acacia Project, Serbawangi Food for the rice, together with MADA and MADA Corp for the rice. And financial support sumbangan adalah daripada MOSTI dan juga daripada Kementerian Pendidikan Malaysia dan juga agensi yang terlibat dalam dana untuk pengkomersialan iaitu Platform Ventures dan juga MTDC ya. Juga masih memberi sumbangan wang. Dengan itu saya ingat kita dah sampai akhir. So yang berbahagia Profesor Datuk, demikianlah sebahagian daripada catatan perjalanan akademik saya selama 20 tahun di UKM. Okay. Dan ketambah 11 tahun di Frame. Pendidikan itu adalah bersambung. So, sebagai tanda penghargaan dan juga terima kasih, saya sudilah kiranya yang berbahagia Profesor Dato' Insinyur Dr. Muhammad Marzuki menerima buku syarahan saya.
Sebagai tanda penghargaan Fakulti Sains dan Teknologi terhadap Pemidato, manajemen jangkauan yang berbahagia Timbalan Naib Chancellor Hal Ehwal Akademik dan Antarabangsa diiringi yang berbahagia dekan untuk menyampaikan cermata kepada yang berbahagia Profesor Dr. Wickneswari Ratnam. Majlis dengan segala hormatnya mempersilakan. Mengumumkan perarakan keluar Ketua Timbalan Naib Chancellor Hal Ehwal Akademik dan Antarabangsa. Seterusnya, hadirin dijemput menikmati jamuan yang disediakan di kafe Canseloria serta melawat pameran di Perdana Canselori. Untuk makluman hadirin, upacara boleh di luar bilik Senat ini. Sedikit penghargaan dan terima kasih kepada semua yang meraihkan upacara perdana yang penuh fenomena ini. Sekian, majlis mengundur diri.